Hey everyone, so I know we couldn't meet live in person this week, so I wanted to go through a couple exam review questions and stuff that I had in mind to try and help you guys be better prepared for our exam this Friday. Again, the exam 3 review will hopefully be back live over Zoom, so you guys can ask questions live and can get more direct things answered. I only had one question for this segment, which I'm hoping is a good thing, and that means you guys understand primary producers and our invertebrate chapters pretty well. I know it can be challenging stuff, but I'm going to go over what Jordan sent in, and hopefully that will give you guys a little more perspective on our invertebrates. And I'll go over a couple more important things that I found interesting, just giving you guys a quick snapshot of exactly what to expect on the exam, kind of like what we did last time. The Jordan specific question was trying to understand a lot of the evolutionary advancements in the invertebrate phylogeny. And of course, I can't go through all of them, but there's a really great chart that Dr. Meadows put in at the very beginning that kind of goes through and tells you some of the distinctions between a lot of these species. For example, if we start down here at this common ancestor, you'll see that everything branches off from there from our chordates, which we'll get to in a bit. I believe that is our next two lectures are our chordates. And we go all the way back to the periphery, which are the first invertebrate group we looked at, which are the sponges. So sponges are the only invertebrate group that does not have a tissue. So if you remember, they have, their osteopores and a bunch of other quote unquote organs that are just cells. They're not actual organs, they're no tissues, but they perform functions like an organ would. So, the ostia is where water comes out of. So that's the osculum, the hole in the middle. The ostula is where water comes in. So they don't actually have tissues or organs, but their cells form functions that would kind of mimic those that an organ has. Next you have Cynaria, which is tissued organisms, and they have radial symmetry. So these are your anemones and a lot of your sponges and stuff like that. And that's why you get broken up into the Cnidaria and the ctenophores, which are, your comb jellies. And you have to remember that comb jellies are not actual jellyfish, because jellyfish are in the Cnidaria phylum. So comb jellies and jellyfish are two very different things. And those ciliary combs are what makes comb jellies, and what separates them into a distinct phylum. So now we're going to go into organs that have bilateral symmetry. So everything on the right of the ctenophores have bilateral symmetry. And that is the platyhelminthes, which are the flatworms, and my personal favorite invertebrate group. They engage in penis fencing, which is when two hermaphroditic individuals, which they're all hermaphroditic, which means they have male and female parts. They will go and fight, and the winner will inject the other one with their penis and to inseminate the other, and the victor can go off and go and live a carefree life with no burden to go and raise young, while the loser has to go and now try and support a pregnant lifestyle and support the young after they are born. They do not have a body cavity. They're really, really incredible creatures. So if you're interested in invertebrates, I encourage you to start there because I love them. They're really fascinating. Then we have Nemertia, or the ribbon worms, which can be really, really huge, and they do have a body cavity. So again, everything on the right of Platyhelminthes has a body cavity. And they have a reduced proboscis, so they don't actually go and stick out and try and grab prey. So that is the big distinction between a lot of the other organisms to the left of it. And the proboscis is reduced and it is present, while a lot of the other ones have either an extended proboscis or they do not have a proboscis at all in their body cavity. We'll go into the mollusks, and there's a bunch of different subclasses in the mollusks, such as the bivalves, that you're going to have to know. But the important distinction here is that they do have a ventral nerve cord or a ganglion, and they have a foot and a mantle. So the foot is what I'm going to focus on here for time. The foot is a muscular foot, which is used for motion. It's a big meaty part. That's what you eat. And that is used for locomotion and moving. We'll then go into the organs that do not have a foot or mantle, and we'll go into the airworms, or Chaetoganatha. These do not have segmentation, which is something that you will see in the annelids, which are the segmented worms. There's a ton of different annelid species. I can't go in too much into depth with them just due to time, but they don't have jointed legs compared to the arthropods, like your crabs, which do have jointed legs. So now that we've gotten past those organs with a ventral nerve cord or a ganglion, we're going to go to the ones that are either not there or not ventral. and ventral means on the underside. So we're going to go into Phorgonia, which are something that I don't believe we actually talked about. I might be wrong on that, but I do not remember talking about them. But they are lophophores. So this is, oh, that's right. These are our lophophorate worms. So we're talking about the lophophorates. There is one that has a true circular storage system, which is the Phoronidia. And then we have two that do not have a circular storage system, the Ectoprocta and the Brachiopoda. So the difference between these two are the Ectophyta are colonial and the Brachiophyta are non-colonial. So think about coral. There is colonial coral and there is, the big groups, and there are some independent like coral fans. That's the way I always tried to get colonial and non-colonial separated. So you can think about it like that. When I go to the non-lophophorate worms, Echinoidea, the sea stars, sea urchins, stuff like that, they have no gill slits. They are the ones that have a water vascular system. The sea urchin has the Aristotle's lantern, which is mouthpiece. So there's a lot of different things that go into the Echinoidea, and I think that's a phylum that you can spend a lot of time on. Again, chordates are the ones that you are not going to have to learn about for this exam. You will next time though. But the hemichordates are basically just chordates with no notochord. And that is a brief, very brief overview of phylogeny. And hopefully that helped clear things up a little bit. I'll go over a couple more things in detail when I go over each specific thing. If you can see up at the top, I have our 4 lecture slides pulled up just so I can kind of go and show you guys the visuals that Dr. Meadows provided, because I know I'm a visual learner. And we'll go from there. And I'll touch on a couple of these things again. But I want to start off our lecture with the microbial world and talk a little bit more about some of our primary producers. Most importantly, this statistic here, 98% of all marine biomass is marine microbes, which is really kind of crazy to think about. If you go to talking about our first question here that Dr. Meadows poses in our module 7, write down 5 marine animals. We could translate that to 5 marine organisms. Chances are you're not going to think of phytoplankton. You're not going to think of a lot of our microbes that are in the ocean. They make up 98% of our biomass, more than those 2-ton whales or massive sharks. They make up a lot of biomass, and that's why they're so important. And they are really great, producers of chemical reactions. They undergo a lot of different chemical changes to the ocean water and provide a lot of food to other organisms. So marine microbes are super, super, super important. So as we keep going down, it's important to note that viruses are not alive. In turn, they are not actually going to go and be part of a domain. So no Eukaryota, Monera, or Archaea. We're now going to go into a couple little bits about our algal diatoms, which are these really awesome, I like to call them art pieces. I mean, we did watch a video all about diatom art, and that's really all they are. They have a silicous ooze that is embedded around them, which becomes that frustal silica shell. They also have diatomaceous ooze, which is a sediment that gets produced. So because they are diatoms, when they become the sediment, when they die off, they become diatomaceous ooze, which makes sense. They're diatoms that are in the sediment. So that's what they're called. Biogenic means it comes from a living organism. So that's a little bit about diatoms. And you'll see here a good example of the frustal. You have the bigger frustal here and the lower one here. So they lock into each other almost like a Tupperware lid is how I like to think about it, with the lid kind of overlaying and grasping onto the lower end of the bowl. So that's how I like to envision the frustal, is something larger kind of grasping onto something smaller, encapsulating the organism inside of it. Diatom art, love diatom art. Here's how to do it on your own. Dinoflagellates are the cause of red tide in the state of Florida. So these are a very important organism. They do have that cellulose cell wall, just like you'd find in plants, but remember, these are not plants. Like I mentioned, they're responsible for some red tides, and they are planktonic, so they're very small, and they do often eat phytoplankton. So they are zooxanthellae, and we'll go, oh, excuse me, let me back up. They do not sometimes eat phytoplankton. They're zooxanthellae, which means they're in coral. They are the symbiotic partner in coral. They are what produce the sugars through photosynthesis for the coral to eat, while in turn the coral gives back protection in terms of a home. Bioluminescence is really awesome. If you're able to, I know on the West Coast, there is a bunch of bioluminescent diatoms that sometimes arise. I was really hopeful to go out this summer to go see some, but never happened. So I'm really hoping to get an experience there. Foraminifers are important, but for the sake of time, I'm going to pass on them. I'm going to keep going into the fungi. So fungi are super important. They are really good at being a symbiote. So they are going to decompose. They're going to live on the roots of like a mangrove tree and help break down dead leaves and stuff like that and contribute to the nutrient pool that comes into contact with those mangrove roots. Dr. Meadows released this really good study resource. So when I took this class, I did use this a lot and I feel like it helped a lot. So table 5.2 in your textbook, Castro's table is definitely very, very good and I'd recommend studying it. And that is the end of our first lecture on the microbial world. It's very important, but there isn't a ton of true meat of content like there is in our invertebrate section. So hence why I spent a little bit less time on these two. That doesn't mean it's any less important for the exam, but I feel like this stuff is a little easier to grasp than some of our later concepts. So we'll now go into our multicellular primary producers. So seaweeds and plants. Giant kelp, if you come to the guest lecture that I did a whole guest lecture on giant kelp. It's really fascinating. They have a lot of biological effects. They have a lot of ecological effects as well. They're super, super, super important. Macrocystis pyrifera is its scientific name. And if you want to know more about it, feel free to message me or just come in and do a little bit of research on your own because it's really interesting. Sargassum is something very important. Sargassum is a seaweed that is kind of just floating in the ocean. It is a green algae. So that is the same kind of thing as the giant kelp. And it is used for turtles as almost a nesting ground. So they are born up on the beach and then they will go and travel all the way up to the Sargasso Sea to kind of grow, use some of the resources there as food. And there's a ton of different organisms that live within the Sargassum and it's very, very, very important. I definitely recommend rewatching the Sargassum Sea video for the exam. I feel like it'll give you a good overview of exactly why it's important and really connect with you, just kind of understanding that, yeah, this macroalgae is very, very important for oceanic functions. Same thing with this chart. This gives you a very good overview of it in case there's a question on the Sargassum Sea. This chart will give you a lot of the ecosystem services and the ecological significance of the Sargassum Sea. This is a really awesome video done by a UF student, like Dr. Meadows mentioned. This is also a good one to watch if you have the time. I would recommend watching the Sargassum Sea ones over this one, just because this is more of an educational one compared to this one, which gives more information about the Sargassum Sea. But this is definitely a good one to watch if you want to rewatch something. So we'll go. I use this chart in my guest lecture as well, and I feel like it's an important one to know. So the thallus is the main branch of the giant kelp or a macroalgae. You have these nematocysts, which are gas-filled bladders. So these nematocysts are filled with a bunch of different gases, and they help keep the blades afloat and kind of keep them rigid a little bit. And that'll help with photosynthesis. So as you can see here, it's kind of branched off at the surface, so it can stay underwater, stay hydrated, and sunlight can hit each leaf. So it helps keep everything separated. The blade is what I referred to as the leaf. I should not have called it a leaf because they're not true leaves. Macroalgae are not true plants because they don't have true roots, they don't have true leaves, they don't have a lot of the other characteristics that a plant has. So in turn, macroalgae are not plants, and that's something that you should definitely know, whether for the exam or in general. It's just knowing that algae and plants are not synonymous. The blades are the leaf structures, which is super important. The stipe is like the stem that kind of comes and connects the nematocyst and blade. It's the best way to explain it is it's like a stem. The holdfasts are like the roots, except they don't absorb nutrients. That is a very big thing, that the roots of the giant kelp, the holdfasts, do not absorb nutrients. Their only goal is to remain attached to the bottom, to whatever the substrate is. So that is the goal and the only purpose of the substrate, of the holdfast, to attach to the substrate. Knowing your types of macroalgae is going to be super important and how to identify them. You can kind of tell this one, you know, it's red algae. Green algae, kind of same thing. Brown algae, we talked about sargassum. This is a sargassum plant right here. So you should be aware this is a brown algae. And then you get into something like this, and it's hard to know, but I would definitely go and review your play posit question on how to identify this, because it looks red, it looks brown. So what could it be? I definitely go and see and revisit exactly how you can differentiate between red, brown, and green algae. And I know Dr. Meadows put a play posit question in there, so I definitely go and figure out exactly how you can identify them. The economic importance of algae is huge. There's so many different things that can go about it. Algin is probably the most popular thing. It's in shampoo, it's in toothpaste, it's in a lot of ice cream. So algin is super, super important. It's found in brown algae, and that's why giant kelp and stuff is a very popular commercial product. The other two, carrageen and agar, are very, very popular from red algae. Carrageen is something you'll again find in stuff like ice cream, and agar is what you are commonly going to see in your agar plates for a biology lab, because I know we do have a lot of biology kids in here. Canning and low calorie foods and stuff like that. It is a good laxative. So it is something that you'll see in a couple of products that we consume as well, but is really a great spot for biological research. That's an agar gel. Biofuels is really interesting too, and then marine plants. So seagrasses are one of the few truly marine plants. Again, they are fully submerged, but they're not grasses. They're part of the lily family, which I found really interesting and I didn't know. These rhizomes are horizontal roots under the sediment. So you can kind of see how they're kind of branching off here. It's a big horizontal root. So that is what the rhizome is. And they're very productive, and they're also a very, very important nursery habitat for a lot of species. So think about like a mangrove forest. We are commonly associate those, as we talked about, last lecture as well with the fungi, that they are very important to have in a nursery setting. Seagrasses are the same way. They're very, very, very important nursery habitats for many fish. Salt marsh plants are another one. Spartina cordgrass is one to know. They're not fully submerged, and you'll see them along the bank, and how they deal with salt. So we learned about mangroves. I guess we technically didn't yet because mangroves are next, but we learned that mangroves have a lot of different ways to deal with salt, but salt marsh plants only have one specific way that was mentioned in lecture. I would go and try and revisit that and figure out exactly what they do to deal with salt. And I do the same thing with the mangrove species. How do mangroves, which live in salt water, deal with salt? Each of the three different mangrove species deals with salt a little bit differently. So I would go and try and determine exactly how they deal with salt. I would also see if you could identify the three different kinds of mangroves and the characteristics between them. So we have red, brown, and white mangroves. Dr. Mills went through those a little bit in the lecture, so I would definitely go and try and figure out exactly what the differences they are. If you go to the beach in, like South Florida, you'll see a lot of floating propagules, which are those big long seeds in the ocean. I always had fun just kind of like throwing them back. They go and they kind of float in the ocean, and then when they wash up on shore or they get this cemented into the sediment, they'll go and try and start growing a new mangrove. So that is how they reproduce, is by dropping those big long seeds into the water and having them float away. I believe that's the last slide, but this no frost is super important, and you can kind of see a boundary between the mangrove forest and a temperature line. They can't go very far north simply because they can't deal with cold weather well. But with global warming, they've begun to push up north farther because now more area is becoming suitable for them to grow. And in turn, they're kind of forcing out some of those temperate species that currently live in those areas. So it is super important to realize that. They all have analogous adaptations because they're mostly unrelated species. And that's something that's super important to realize, that they're separate species, but because they had the same environment, they grew together and in turn became clumped together. So I would definitely go and try and understand what those analogous adaptations were from lecture, and understand exactly what analogous means. And again, that means they grew together based on similar conditions from being unrelated. So that is the end of our primary producer section, both multicellular and microbes. And I guess microbes doesn't always have our primary producers involved in them. We do have some non-photosynthetic or chemosynthetic ones, but that is our summary there. And now we're going to go on to the invertebrates. Again, your 5 marine animals, you most likely might think of a shrimp or an octopus, but you're going to think of true chordates, not invertebrates. So we're going to go and try and focus on some of these organisms, the sponges, the arrowworms, other organisms like conchs or whelks, other organisms that are super important, but you might not think of right off the top of your head. So animals and symmetry is super important. Symmetry is something very self-explanatory. So for the purpose of time, I'm going to skip past that. And again, this first lecture is now these 8 sections. So I'll go through a couple of them very quickly. Yes, this is not a sponge you'll find in the ocean. They look like this. They're the ones you see at like gift shops and in those old, they're like loofahs is probably the best way to explain it. Again, they have no organs, no tissues, and they're asymmetrical, and they have a cellular level of organization, but they do have a lot of structures that kind of act similarly to organs in their cells. So here you go, you have, a widening cell, a porocyte, a pinocyte with some spicules for defense, and a collar cell. So those kind of work together all together, almost like an organ would, but remember it is not an organ, it is a tissue, and it's not a tissue, it's just cells working together. So this is not a tissue or organ. That's something important to note. I note exactly what those ostia and osculum do. So again, ostia takes water in, osculum takes water out. And I believe we also watched a video on when someone squirted like that yellow dyed water. You could see it kind of just get sucked in and pushed out of that giant sponge. So try and know what the porocyte does, the amoebocyte, the pinocyte, and the spicules and the collar cells do, as well as the ostia and osculum. And that'll be a very good start for exactly what you do or what to know about the sponges in phylum Porifera. The flagellum is also important for food movement. So I'd also know about the flagellum and that choanocyte. So if you can do that, you will be set for sponges pretty much. Also know what happens if you centrifuge some sponges. They actually start coming back together, which is crazy to think about. So that's really, really interesting. They're filter feeding and suspension feeders. So that means they take that water in, they filter it out, and they push it out through the osculum up top. So let's go. Sponges, know about the reproductions. They can either bud and break off and create a new sponge, kind of like how we saw with you centrifuge them. If you separated those cells, you'd be able to go and create 2 new sponges. And then sexual reproduction can be done via broadcast spawning and picked up by an egg that also got broadcast spawned and spread out. You don't need to know too much about Dr. Meadows, invertebrate zoology professor at UNC Wilmington, but his research is pretty interesting and it kind of relates back to chemical ecology, but it's something interesting to note about sponges. Human drug applications, there's a ton of them. So HIV drugs are used or being created from sponges, which is really interesting. And hopefully they can go and expedite that process. I know they're going and making a vaccine for HIV using the mRNA technique they did for the COVID-19 vaccine, which is really interesting. There's also been done to treat herpes, and there's an anti-leukemia drug that's been out for, at this point, I believe that's over 50 years now. So there's definitely a lot going on in terms of sponge and chemical ecology and human uses. So the first marine drug for cancer treatment was done from an organism in the phylum Porifera, aka the sponges. There's a ton to know about them, but again, for the sake of time, I'm going to go on to phylum Scenaria, which are the combs, the corals, or not the combs, the corals, the jellyfishes, and other organisms. You should know the medusa and the polyp stage. So the medusa is what you see in like a normal jellyfish, while the polyp is more of like a coral structure or sea anemone. I would go and understand the difference between those two and when in each life cycle they exist. Know about the nematocysts, which are those stinging cells. And then here's a couple more phylums that go about it. The life cycle is not super important, but you should understand the basics of it. Just again, knowing when the polyp and when the medusa stage exists. Hydrozoans are kind of important as well. Just know that they are those drifting colonies of polyps with those nematocysts attached, and an example would be the Portuguese man-o-war. So these are colonial species. They're not an individual jellyfish. Scyphozoans are those huge jellyfish you'll see. This big top layer of the medusa is called the bell. And so it actually kind of just compresses and contracts almost like you would like a bird flapping their wings in and out, kind of compressing and contracting. Cubomedusae, the box jellies, they look like a square box cube, hence why it's Cubomedusae, and it's a box jelly. Kind of self-explanatory. Very powerful stinging cells, nematocysts, so good to know. Anthozoans are corals and sea anemones. So again, they don't have a medusa, so they are only in that polyp stage. A lot of them are colonial, but there are some singular ones. And the corals secrete that calcium carbonate skeleton, and they do have that zooxanthellae, which, if you remember from earlier, is the dinoflagellates in the primary producing section. Here is Dr. Meadows diving underwater. Again, if you haven't started diving yet, go do it. It's awesome. But we're going to go on and continue into actually watch this video here about the spawning of corals, understand like the importance of the research that Dr. Meadows did. I think it's really fascinating and something you should definitely consider as you go in and study. So we're going to go here and talk about the ctenophores or the comb jellies. Remember, these are not actual jellyfish. And the big thing is they have those 8 rows of ciliary combs, and they have biradial symmetry. They are not radial symmetric. And then the colloblasts are the sticky parts of the tentacles used to capture prey. So that is something that is super important, and they're mostly planktonic. So we think of jellyfish as these huge things, but comb jellies, because they aren't really jellies, are not always that big. So now we're going to talk about some of those biologically symmetrical worms, which is most of them. The cephalized anterior means that there's nerve endings by the head. And that's something that's very important that has come about in these phylums, is that cephalization of the head, which allows more complex behaviors, more complex anatomy, and everything becomes a little more complicated and able to go and do more functions. So as we go on, we're going to talk about those flatworms, the platyhelminthes. These are the most basic flatworms or basic worms at all. They do not have an anus, which means it comes in and out of the same hole. So that's not always fun, but that is something to note about the platyhelminthes. I mentioned in, I believe, a guest lecture that they can go and if you cut off from a certain species 1/274th of that organism. So think about like taking 1/274th off of you, however much that would be. That might be half a finger at most. And that can grow into an organism that has the exact same memories as the original one. Just by cutting off half of your finger, it can regrow another one of you with the exact same memories you did. It's really fascinating. And that's one of the reasons I love these flatworms. They're really interesting. I love them. Might have an exam question on that. I'm not saying anything, but yeah, I've already talked a little bit about penis fencing. This is important. I know about penis fencing. It is a very important characteristic of the tubularian and platyhelminthe species. So understand exactly what that is and how it happens. I'll pass over flukes for a second. Tapeworms are important. Just know that they are parasitic and they live in the guts and take away a lot of the nutrients from organisms. Nemertia, like I mentioned, have that long proboscis that you just go and stretch out and go and wrap around, in this case, a polychute worm. So they do eat their prey, and they do have a complete digestive tract, which means unlike the platyhelminthes, they have an anus. They also have a circulatory system, unlike the platyhelminthes, and they are potentially the longest animal, because when you stretch one out, they get very, very long, and some say it might be larger than a blue whale. So we'll see how that actually is. We haven't found definitive proof yet, but it is most likely the largest invertebrate in terms of length. Overall, don't know yet, but it is possible. He's Dr. Meadows' husband with the ringworm running by his feet. Anaxasis is definitely something I would go and try and learn about. Know each of these steps. I'm not going to go through it now just for the sake of time, but I would definitely go through and understand how this occurs, why this occurs, the larval stages, and really kind of go into exactly what occurs for these roundworms in the phylum Nematoda to go and become parasitic and get inside the digestive system of a human. The arrowworms or Chaetognatha are a smaller planktonic predator species that kind of look like an arrow, like you can see here. Bilateral symmetry is very, very easy to see in this diagram, which is why I very much like it. You can draw a line almost right down the middle of it, and you can see that it's symmetrical on both sides. So it's really interesting. Again, there's only 100 known species, so there's not a ton of them, but we are discovering new species every day. So it is exciting about that. The annelids are hugely important. They have a closed circulatory system, and they are the polychaete worms. I know exactly what the parapodia and setae are. Those are important anatomical features. And again, I'm not going to go and talk about it for the sake of time, so I don't waste your guys' double speed, because I'm sure you guys are time. But definitely understand parapodia and setae and what they are. Here's a really good depiction of some of their anatomy. We can kind of go in and get the answers to those questions that I mentioned earlier. Same here, these are some drawings. I'm not a huge fan of drawings, but like this one here is incredibly detailed. So you can get a lot of good information from that. There's a couple of specialized ones. I wouldn't know too much about them, but understand that there is some specializations that do exist. And glowing. Glowing is super interesting. Luciferin and luciferase are the chemical and the enzyme that it uses to go and create that glow. That's one of my favorite facts. I love luciferin and luciferase. I think it's really interesting. So I would know about those two and how these annelids grow or glow, excuse me. Hydrothermal events are important. Just understand some of the adaptations that the beardworm has for hydrothermal events. I think that is something important to know because it is a very different ecosystem down there. Important to note that echiurans used to be their own phylum, but have now been grouped in with the annelids. So I definitely know about that. And someone like this proboscis here, I'm trying to understand exactly, what it does with that non-retractable proboscis, why that is important, and some of its characteristics as well. Same with phylum Staphypula. Staphypula has been taken into the annelids, and know a little bit more about its characteristics as we get to the end of this lecture. So we'll go to our final lecture on marine animals without a backbone. Let me zoom out a little bit so you guys can see better. Here's that phylogeny again. Again, that's probably the best you're going to get in terms of just overall organization. And we're going to talk about the mollusks to the hemichordates. Chordates, again, that'll be a little bit later on, but based on what we talked about here, I'd still know a little bit about them. Mollusks, super big snails, clams, octopuses, squid, lots of them. That mantle is what secretes the shell. So that is a calcium carbonate shell that goes and gets secreted by them. Their body plan is greatly modified. That means there's a lot of torsion or movement within the body. So you see it all coiled up, and this is a snail, it's all coiled up in the snail shell. So that is what that greatly modified body means. It's a lot of just movement of parts and different organs kind of modified to do different things. The radula is that ribbon of teeth that's used for feeding. A lot of it is a scraping radula, which means like in snails, it kind of goes along and scrapes off algae and other things from the bottom. It's modified in predators, whoops, I apologize. There we go. It's modified in predators, so it doesn't scrape off the bottom, but it is still present. The gastropods are what you're going to get in terms of like a lot of those limpets and abalone and snails. So it is the most common and most varied group. Here's that scraping radula to get algae. So I know a little bit about the coniverse ones, the deposit feeders, the nudibranches. Nudibranchs are awesome. They are really interesting to see underwater. So if you have the opportunity to go see some, I definitely would recommend. So this is a good little summary of the class gastropoda, excuse me. The bivalves are your mollusks, your clams, a lot of your other, I can't even think of any other ones right now. Mussels, oysters, it's on the screen. But they're laterally compressed, which means they all kind of squish down into that body. You have two valves on the shell, your left and right. The adductor muscles are what closes the two shells. So those are when you open up a shell and it's kind of empty and you see that little like divot in the corner, that's actually where the adductor muscle is. They are adapted to no head, no radula, so they don't actually have either of those, but they are very cephalized still. So we'll go a little bit more in depth about it. They use their muscular foot to burrow, and they attach to rock with byssal threads. So that is something important to note. They're almost like hold fast, where they just grab onto a surface. Some of them have zooxanthellae, which is something to note. And again, zooxanthellae are dinoflagellates. So that's something important to remember. Cephalopods are your very cephalized, hence cephalopods, organisms. So they have chromatophores and dermal photoreceptors, which allow them to change color based on their environment. Ink sacs are used to confuse predators, and jaws exist. So you might have seen videos of like an octopus that is able to fit through a really, really small hole because the jaw and that beak is the only things that are truly not flexible. So as long as a hole is bigger than their beak, they're able to go and fit through that hole, which is really interesting. Their muscular foot is modified in suckers, and it's important to note that there are only 7 legs for an octopus. One of them is its penis, and it's used to inseminate females. And it can oftentimes come off during reproduction, which is kind of crazy. I know about the siphon and how it is used to kind of spit water out and help the octopus or squid get pushed away. True fact, I love true facts, always interesting. Here is a good depiction of a squid with the pen, which is kind of like that support structure. If you ever are preparing one without going and buying it already pre-done, make sure you take the pen out. It's not fun. And that is what the shell is. So if we go down here to like the nautilus, that big shell is reduced to this, and they're in the same class, which is really interesting to think about. They have 8 arms, 2 tentacles. So these tentacles are the big ones, the arms are the little short ones, and they are adapted for very, very fast swimming with the in-current, excurrent siphon. Nautilus and cuttlefishes are another one just to keep in mind and understand a little bit about their anatomy from what we talked about in lecture. Here's some other mollusk classes, just know general characteristics about them. So the phylogeny will go into the arthropods, so like your crabs and other organisms like that. The jointed appendages is a very big jump that occurred in this phylum. So joints did arise here. They do have legs and a more complicated mouth part, and their bodies are segmented, just like the worms we talked about a while ago. Chitin is what the exoskeleton is made of, and it's for protection, support, flexibility, and muscle attachment. So chitin is a very, very, very valuable resource that these arthropod species use. There's a ton of diversity in this species, but there is a lot of marine ones as well. You're not going to find a ton of either freshwater or land arthropods. A lot of them are marine crustaceans. The crustaceans are a lot of them. Again, roly-polies are crustaceans. They're the ones you're going to find on land. That chitin skeleton exists still. So that's something important to note. They have an open circulatory system, which is not something we've seen before. I mean, it is something we've seen before, but if we go back to those like ribbon worms that had closed one, that is something that did not get evolutionary translated up to the crustaceans. So that's important to know. They're not hermaphroditic. So again, hermaphroditic, like the platyhelminthes, means 2 organisms have the same sex. It's actually really interesting. There is I believe it's like 1 in 100,000 chance that a lobster is a hermaphrodite. And there's a museum up in Massachusetts where the lobsters that are hermaphroditic are put on display because they actually are a different color. They're white, some of them are blue, and there's a lot of, I believe it's actually called like the lobster rainbow or something along that line, because there's just so many different colors of lobsters because they're hermaphroditic. So it's really interesting about that. Here's some more arthropods, the crustaceans. You know the difference between a copepod and amphipod, which we learned a little bit about with our guest lecture from Brittany Cummings, and then our isopods. So these are some interesting creatures. Just know the difference between them, between the copepods, the scuds, and the isopods, and maybe some deep sea giganticism with these isopods because they live at the bottom and why they're huge. Here's some barnacles. They have the largest penis to body ratio. I believe it's like 8:1 or 9:1. Yep, 8:1 right here. So that is something that gives them an evolutionary advantage to go and reproduce while being sessile. Krill, understand the ecological importance of krill. That's going to be huge, and I believe there's a video on it as well. There is not. Yes, there is. Yes, there is. Yes, there is here. So understand their ecological importance. That's super, super important. I'd watch both of these videos. Stomatopods, also important. Understand how the mantis shrimp uses it. I'd watch these videos as well. I think this is very important. The arthropods, now we get into the decapods, which means 10 legs. So these are your lobsters and other organisms like that. Hopefully, you know their economic importance. You know, they're a huge food source. The chilipeds, know that those are those big grasping claws, and those are modified. The pteropods are the other 5 legs, or 5 pairs of walking legs, or 4 pairs, excuse me. There's 4 pairs because the fifth one are those chelopeds. The maxillipeds are the 3-part mouth, and that is what they use to eat. And so just know the pleopods slash swimmerets and what they are used for, which is, again, for motility and swimming. Shrimp and lobsters kind of fall under that same thing. Hermit crabs are not true crabs, interesting to note. And you have these little small ones that you'll see versus the giant coconut crabs, which are both crustaceans and hermit crabs, which is interesting. Brachyurans are your true crabs, which are really cool. I love brachyurans. Abdomens are tucked under the cephalothorax, and it is important to note the difference between a male and a female crab. So if you can identify that based on this image, then I think you're in good shape for the exam. Here's some larvae. I know about the larvae, just know the difference between them, which organism has what kind of larvae, and that's something that was in our Smart Book assignment. So that's something I definitely go and revisit. So we'll go, these are some of our other ones. So horseshoe crabs, we've already talked a little about. Sea spiders, I don't believe we have before this lecture. So I definitely go and understand a little bit about some of these, and the importance of blue blood is super important. Even in developing the COVID-19 vaccine, horseshoe crabs were very important. And that's something we discussed here about the lysate, amoeba LAL. Here's our revisiting the phylogeny again to kind of talk about the lophophorates. So it's that unique structure that kind of sticks out that is used for feeding. That is what the lophophorate is. So we have those 3 phylums. I know the difference between the 3, and just the lamp shell, the Brachiopoda, the Phoronida, and the Bryozoa are the 3 groups of organisms within the lophophorates. I know each of the 3 and how to identify them visually. Echinodermata are, we're getting near the end, thankfully, because my mouth is tired, but these are our sea stars, our sea urchins, our sea cucumbers, and a lot of other organisms there. You have the madreporite here, which is where the water vascular system really starts. And so I really know the purpose of the water vascular system that controls the tube feet, and the tube feet are used for movement. So I know that. Regeneration, so you probably know that if you cut off the arm of a sea star and take off some of that central ring, you'll be able to go and make a new one. So you have oral and aboral side. So the oral side is the underside, the aboral side is the top, because their mouths are at the bottom. Sea stars are in the class Asteroidea, which is really nice that sea stars and asteroids kind of get that space vibe. Sea stars are super important. Here is, I believe, the crown of thorns sea star. And so these are your typical example of what you'll see in the Echinodermata. Little stars are those big, long, feathery ones that have that central disc that is very easily to see, which you don't always see in sea stars. Here are sea urchins and sand dollars. So they have that big, long exoskeleton that's hard. They have that Aristotle's lantern, which is a big mouthpiece at the very bottom that is used for feeding. So I definitely know about that. And I believe that is talked about on the next slide. Yep, right here. And you can kind of see a really good example of what the Aristotle's lantern is here. And this was actually described by Aristotle, which is cool. I've seen these in person. These are pencil urchins. They're really cool. They do not hurt because I accidentally bumped into one while diving without realizing it. But pencil urchins are really awesome. I love pencil urchins. Sea cucumbers can eviscerate their guts whenever they're threatened. That is the big thing to know about them. They're elongated orally to aborally, which again is from the mouth to the gut. So you can see it's very stretched out. And so that's really the important thing about sea cucumbers. Feather stars and sea lilies, there's not too much to know about them, but they do incorporate a lot of the other features of the echinoderm. The hemichordates are also deuterosomes. I know what a deuterosome is and understand the 2 chordate characteristics. I would know all of them by the end of this lecture, which we're going to talk about. So they do have the post-anal tail in some species and the gill slits. And gill slits is something that we saw on that phylogeny at the beginning of the lecture. And here we go, the 4 chordate characteristics: that dorsal hollow nerve cord, that nodal cord, the gill slits, which are also called pharyngeal slits, and the post-anal tail. I did say 1 and 3 together because in my opinion, it's very easy to remember dorsal hollow nerve cord and nodal cord because they sound very similar. To have those back to back was helpful for me when I memorized them. this is something that all chordates will have. So we're in the phylum Chordata now. So yeah, you would not expect these to be chordates, but they are, because you can still lose characteristics and still be a chordate. So these have all these characteristics when they're younger, but they do not have them when they are full-grown adults. These are still chordates. So these sea squirts do have this extranth siphon and other things here. And those tadpole larvae, like I mentioned, have all 4 of those characteristics. They're the only sessile chordates, and this tunic is what protects them on the outside. It's oftentimes made of mucus and other protective structures. The salps are these big, long, I really want to see these in person because they're like those bubble wrap packages, those big bubbles that you get sometimes that are just super long, and they're not the single poppable ones, but big, like 6-inch by 3-inch big ones. And I really want to see one in person. But they're super important carbon sinks, and when they die, they sink down to the bottom as detritus, or what's also known as marine snow, and they're super important for the benthic food web in the ocean, open ocean. So they move water in by taking water in one end, the incurrent siphon, all the way out of the other end, which is the exurrent siphon, and they are planktonic because they cannot swim against the current. Larvaceae is another one of our chordates. Here is a really awesome depiction here of how water moves in and out of the organism. They make a house out of mucus, and this is another super important part of marine snow. It's a very important carbon sink, and that will sink to the bottom often because they do get rid of that house very, 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 very often. The lancelets are a really great example. This is what you're typically going to see out of a chordate to get a lot of those 4 characteristics. They keep them all throughout life, which is not often seen. So you can see the notochord, which is this red one here, the dorsal hollow nerve cord, those gill slits or the pharyngeal slits, and that post-anal tail. Here's the anus, here's the tail. So that is the important things about lancelets. And that is the end of the lecture. So hopefully you guys got some good information out of that. I know it's a bit long, and I know you guys are probably going to double speed it, hence why I tried to talk a little bit slow. But if you do have any more questions before the exam, please feel free to let me know. I am here to help you guys, and hopefully this was helpful. And I look forward to seeing you guys all with our next lecture.